Good evening to everyone.
Good evening, sir.
Good evening, sir.
Am I visual to everyone?
Yes, sir.
I hope all the slides are visible, all of you.
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Okay. Thank you, Amrit, for the nice introductions. So thanks for joining today's webinar. On behalf of Heredity Biosciences, I am Dr. Gopal Purohit, CEO and co-founder at Heredity Biosciences. Welcome to all to attend this webinar today. As you know that today we are conducting webinar. Topic is applications of flow cytometry in modern healthcare research, presented by Dr. Arpit Shrivastava, visiting faculty at the School of Biotechnology, DAVV INDOR. The question is that why it is unique webinar today, because we will learn lot of points related to the flow cytometry, related to the modern healthcare research, and our speaker, Dr. Arpit, will brief everything related to the topic. Before starting to this webinar, I am little bit highlight to the topic. Probably everyone attending to this webinar, have heard about this flow cytometry? So, but the question is that what is the functions of this, the relations between the healthcare, disease diagnosis and research perspectives. So the flow cytometry techniques used to detect the major physical and chemical characteristics or characterizations of the population of the cell or particles. In this process, a sample containing the cell or particles is suspended in the fluids and injected in the flow cytometer instruments. The flow is focused into the cells at a time through the laser beams, where the light scattered into the characteristics of the cells and the components. The cells are labeled with a fluorescence marker, so the light is observed and then emitted to the band of wavelengths. 10,000 to more of the cells can be quickly examined and the data gathered to the process of the computer. So flow cytometry have various applications for the basic research, like clinical practices, clinical trials. Use of flow cytometry include cells, cell sortings, determination of cell characteristics, functions, determination of microorganism, biomarker detections, diagnosis of healthcare disorders and blood cancers, measuring genomic sizes also. Flow cytometry has various use of this that greatly impacted the scientific research and the development of the drug therapeutics. In many applications of flow cytometry that have resulted in academic, biotech, pharmaceutical, and clinical levels also. And it's a more advantage also. This advantage have specificity throughout in the many multi-parameters to the flow, to the use of this molecular biology, immunology, virology, infectious disease, and cancer biology also. So this is a little bit brief about this the topic. Our speaker, he is highlighting all these topics. Now, before starting to this webinar and our guest of introductions, I will little brief about this Heredity Biosciences. Heredity is established in 2018. And it's renovated for new structures like Heredity Biosciences in 2021. And it's providing research and training services to hospitals, life sciences institutes, pharma companies, and trusted name among the doctors, research scientists. It is backed by well-experienced scientists and sales professionals with a strong background in the life sciences sectors, healthcare sector, analytical, metabolomic services, of different industries. Our team has a strong standing experience and expertise in delivering quality services and innovative products. So we have a strong team equipped with a National Scientific Advisory Board, national skilled professionals like professors, doctors, scientists in the field of life sciences research, proteomics research, metabolomic research, genetic diagnosis, and bioinformatics also. We aim at delivering high quality services and cost effective solutions using the technology and support of life sciences, research, clinicals, sectors, and industry in the India and abroad also. Now it's time to focus in the company's core activities. So we are focusing to these life sciences as well as the healthcare services and both of these activities. And we are providing for the complete hands-on for this training as well as the research activities for the professionals. Now we will move on the Heredity Biosciences portfolio of services. So we are providing for the genomics, proteomics, metabolomics, and analytical services in genomics. We are providing for the bacteria, fungus, PCR-based microbial quantifications, PCR-based antibiotic resistance gene analysis, microbial DNA barcoding, real-time PCR various services. Proteomics is like protein profiling, post-transcriptional modifications, protein kinases, data analysis, so many things in the proteomics. Metabolomics is perspective like targeted metabolites, on targeted metabolites, and unknown metabolites also provided for that. And in analytical services, we are introducing this month like LCMS analysis, GCM analysis, HPLC analysis, and FTIR analysis also. So in healthcare point of view, we are focused on the research part like clinical microbiology, infectious genetics, cancer genetics, fertility genetics, and conventional PCR-based DNA study to disease diagnosis of the various experiments. So we are focusing not only for this Odisha, we are focusing for the pan India also. On the Heredity Bioscience training activities. So we are providing for the various experimental things as well as for these hands-on experiments. This is provided for the Heredity Biosciences. We have a various portfolio of the training for like clinical microbiology and genetics, genomics and clinical hematology, microbial nanotechnology and genetics, food microbiology and genetics, and various specialized modules. So we are introducing like a clinical embryology, RDNA technology, qRT-PCR, disease diagnosis, histochemistry, and immunohistochemistry. So any life sciences students eligible for these programs. So we have and well-established world-class laboratory available and complete research learning facility, one-to-one given for the students, well-trained and experienced trainer and demonstrator to the individual experiment exposure, publications assistance to the students, but students should be writing to the publication. We are assisting for that how to write it, placement assistant to all the students. Those students are interested. And one-day training, we are inviting all the students, if anyone is interested. So we are conducting for the Saturday one-day training program. Anyone who be interested to join for this program for the high-end experimental series, but that is a complete hands-on. So this 6 to 7 months, Heredity Biosciences conducting so many one-day training programs, completing for the hands-on experimental procedures, and more than 300 students completed over the short-term and long-term internship programs. That is 1 month, 45 days, and 3 months of programs. That is a complete hands-on procedure of the students. These are some of the certificate distributions of students. It is the first time in Odisha we are providing for the complete hands-on procedures. The students should be doing for all these some of the photos and pasted here for the experimental finally certificate distributions the students. So various topics were covered to one-day program, that is clinical microbiology, genetics, hematology, molecular biology. We have tools and techniques of molecular biology and infectious molecular biology and genetics to cover the one-day program. And certificate distributed various scientists and professionals they are given for the final distribution for the one of the students. So this is the one-day program of experimental activities of the students. Some few students are tested here. This is the research activity, laboratory research activities experimental activities going on with Heredity Biosciences and some of the research activities going on. These are these pictures posted here. This is the placement achievements. This is our achievements. So many students of the students have joined from the different organizations, and some of the students are joined from the different fertility centers also. And recently, 2 to 3 students joined for the CIFLA industry. Now, Heredity Biosciences focus into the every time in a research and experimental activities. So we are published more than 25 to 30 publications, and right now, last month we are published. We are filed 2 patents, patents, and 2 to 3 papers right now in the pipelines. Now, it's a time for the introductions of our guest. I'm thrilled to introduce to our incredible guest speaker for today's webinar, Dr. Arpit Shrivastava, who is going to talk about applications of flow cytometry in modern healthcare research. He is a renowned scientist with an impressive 8 years of experience in molecular diagnosis and infectious disease. He is widely recognized as a passionate researcher, making him a truly authority in the field of biotechnology. Dr. Arpit is a proud graduate of KIT University, where he earned a Ph.D. from 2012 to 2017. He has also had the great opportunity to join International Medical University Malaysia during his doctoral program. And I'm also so lucky with work together long year back when I was continued my PhD. I remember one word of Sir APJ Abdul Kalam: failure will never overtake me. My determinations to success is strong enough, and the hard work always lead to success. It proved to Dr. Arpit Shrivastava. In addition, Dr. Arpit is a respected contribution to the scientific community, regulatory sharing and site of expertise. He has experience in Scientist B at Atal Bihari Vajpayee Government Medical College, and extensive experience in the conducting of molecular epidemiology studies, including survey of randomized controlled trials on infectious disease. It is a great and share that he has developed Tagman qPCR assay-based molecular diagnosis for the diarrhea pathogens, dengue plovum, and C. hemonis, and serotypic specific PCR-based diagnosis for dengue. Later, he was worked on the IMBRI impact of analysis of rubella vaccinations on population community in India, and international collaborations project between the John Hopkins University, the World Health Organization, and the Indian Council of Medical Research to estimate age-specific populations immunity to malaise, the rubella virus in India. But Dr. Arpit was working on the scientist at the National Jamalian Institute of Microbiome Disease, adding to the unique perspective and accomplishing innovators to his work. Today, we are so excited to hear Dr. Arpit's thought on the applications of flow cytometry and modern healthcare research. We will be discuss at the latest advancement in the flow cytometry and their potential application in the healthcare research. And we are thrilled to have to speak on this topic. We are looking forward to the talk and the question answer sections, and we are confident that it will be enrich the experience in everyone involved. So let's give to this warm welcome. So this is the team of Heredity Biosciences. They are most expertise teams. They are involved to the research activities, training activities. They are writing for the papers. They are supportive of all these experimental parts. I'm really proud of my teams. They are doing so many experimental activities as well as the training activities also. They are more hardworking to the work. This is a Heredity Biosciences websites, hereditybio.in, and this is the At this portfolio also, we are gathering for the 4.9 rating for this 306 reviews. Right now, we are collected for this. I'm thankful to everyone. Thank you too. Thank you. Now I am called to this Arpit sir. Take the next person.
Thank you very much, Dr. Gopal, for giving me the nice introduction. So you are also the equal partner during my PhD. So you have also guided me a lot. So am I audible to all?
Yes,
Yes,
sir.
sir.
Yes,
Okay.
sir.
So as you have understood this, we will going to discuss about flow cytometry today. So I would like to go first to the slides. So are you able to see my screens? Okay. Hello. Now am I visible?
Yes, sir.
Yes,
Yes.
sir.
Yes, sir.
Okay, so once again, I would like to thank Dr. Gopal for providing this nice introduction. And today we will start the flow cytometry here. Okay, so I'll just quickly go to the slides. Everyone is able to see the slides?
Yes, sir.
Yes, sir.
Yes, sir.
Yes,
Okay,
sir.
good. Thank you. So today we will discuss, as you must know, the applications of flow cytometry in modern healthcare research. So I believe you must be knowing about flow cytometry technique, maybe a little bit basics, or if not, then we will discuss few things. What this is actually a flow cytometer from specifically the PD. Then after the completion of this webinar, what we will learn? Actually, we will learn the principle of flow cytometry, the fundamental parts of flow cytometry machine, how it functions, the result interpretation, the basics, because it takes more time and experience to understand the result completely. But we will see the glimpse how the results can be interpreted and the major applications of flow cytometry in healthcare research. So, what is actually flow cytometry? A flow cytometry is a Greek term. The cyto means cells and meter means measure. So it's a powerful technique that can provide us the information about the properties of cells. On the basis of morphology, cellular properties, we can differentiate cell, and we can also do the cell cycle analysis, because different point in the cell cycle represent the different phenotypic characteristics. So on the basis of flow cytometry, we can differentiate cells, we can sort cells, and we can do the phenotypic analysis. So actually a flow cytometry is an analytical technique that quantifies the frequencies of cells binding to a fluorescent antibodies and scattering light in a characteristic way. It's a precise form. We will understand in detail in next few slides what is the actual principle of this technique and what is the physics behind this. But this is the basic. So basically, cytometry, what does this in flow cytometer? It differentiates cells, cell population on the basis of three things. Number one is size, number two is complexity, and number three is phenotypic characteristics. How? We'll see in next slide. Like neutrophils has a larger, structure in comparison to lymphocyte. The neutrophil is more granular in comparison to lymphocyte. So neutrophil, when it will pass through the channel, it will give a different scattering effect. And lymphocyte when go, that will give a different scattering effect. We'll see that. But the basic is that flow cytometry differentiate cells on the basis of size, complexity, and phenotypic characteristics. So broadly, flow cytometry is used in cell sorting on the basis of size and complexity, cell cycle analysis, in cell viability assay, cell cytotoxicity assay, and sometimes if it is labeled with the antibodies, fluorescently tagged antibodies, it can be used for segregating desired cells on the basis of antigen-antibody interaction. What is the principle of flow cytometry? It's a basic principle, but it's cleanly described here. Is what happened when a particle passes through a laser beam, as you can see in the picture, diagrammatic representation. When a particle passes through the laser beam, it scatters light according to its physical property. For instance, a larger or a more complex particle will cause a higher deviation of light. In case a particle is also fluorescent, it will also imitate a specific light of its wavelength. That will light another higher wavelength that will give a different signal. So if it is tagged with a one different fluorescently tagged molecule, it will get a different fluorescent tag. So in flow cytometry, the optical to electric coupling system is used when the laser beam is used as an optical system, and the electronic system like computer and the different machinery is used to convert this optical signal into this electronic signal. So it's a basic flow chart which includes flow cytometry includes the optical system using the lasers, and the detectors, and the electronic system and computer to read this particular result, which is generated by the different samples by scattering effect. So one important aspect in this flow cytometry is hydrodynamic focusing. It is the most important, the principle for this flow cytometry. What happens? What is hydrodynamic focusing? Actually, hydrodynamic focusing, it is used this term. It uses the liquid or hydro to force the cell into the travel. Therefore, it is called hydrodynamic focusing. If you see this picture, you can see the two different color fluids are presented in this diagram. One is outer fluid, which is called as sheath fluid, and one is inside this test sample. So you can imagine the sample is injected into a one another liquid sample, which is this the hydro liquid or sheath fluid. So what happens if the sheath fluid is a higher rate of dynamics? This internal test sample will be run through this sheath fluid sample. So what happens? The sample is injected into the center of the sheath fluid, which consists either water or the buffer. It will run through the sample, and these cells will be come onto the simple same axis and run at the almost approximately the same velocity. So when the laser light it passes the individual cell. So it can track the individual cell, whatever the cells present into the test sample. So this hydrodynamic focusing is very important to achieve the uniform axis and almost the same velocity of the test sample. So this hydrodynamic focusing, according to its scattering effect, combines the flow cytometry principle. So this is the representation of pictorial representation of flow cytometry, including the parts. The three different basic parts includes fluidic system, which I showed like previous slide, hydrodynamic focusing, optical system, the laser lights, which is important to excite the molecule and its scattering effect. And the third is electronic system, which converts this data to the electronic system and finally display us onto the screen of laptop or desktop. So this is the fundamental part of flow cytometry. This seems simple because I just wanted to explain you, but in reality, it is a bit complicated, just like this. Because we have n number of fluorescent beams or fluorescent lasers, because we want to see the different phenotypic characteristics, size and granularity, and so many things in a single test run. So for this, the different detectors, different laser beams, and different other fluorescent dyes are being used. So it is a bit complicated. And simultaneously, if we want to segregate these things or sort these things, we need to apply this magnetic field. On the basis of magnetic field, these charged particles can be separated. So it is a bit complicated. But initially, when the flow cytometry was started in early 1970s and 80s, it was very simpler form. But in modern world, it is very complicated because it can do a multiple purpose in a within a few hours or few minutes. So it just help us in our research and development. And how this scattering helps us to generate result or interpret results. So in this particular mechanism, the result is being interpreted on the basis of forward scattering and side scattering. So what is the meaning of forward scattering and side scattering? So first we will discuss about forward scattering. So forward scattering is, these are the detectors located within the flow cytometer. These detect the scattered light along the path. As you can see, the first image, first picture, the upper one is the smaller molecule and the lower one is the larger molecule. So in the path, the light is deflected more when the size is more. When the size is less, the deflection will be less. So this is simple. When the scattering occurs along the path is called as forward scattering. This is a low angle scattering because no higher angles is involved in this type of scattering. It is known as FSC. In the second part, when the molecule measures the scattered 90 angles, 90-degree angles, or more higher angles, it is called as side scattering. Side scattering occurs when cells has more complex structure or more granules. So the different granules and different structures deviate the path of light, and it will deflect different so many times within the orbit or within the surface or within the circle. So that's why if more complex structure is this, then the more side scattering will occur. So as you can see the third graph, the larger the size, larger the peak you will find. Smaller the size, smaller the peak you will find. But in flow cytometry, you will find results like this. The first graph is showing side scatter, and the x is showing side is scattered, and y-x is showing cell count. So the higher the cell count is represented the side scattering. These second slide shows the forward scattering and the cell count. It is showing the higher forward scattering and higher cell count. But these graphs individually makes no sense. Once these are combined, side scattered and forward scattered, then only we can get a some important or valuable result or outcome. These results individually does not make any sense. How? So higher side scattering represents the higher cell complexity or higher granularity. Similarly, the higher forward scattering represents the larger size. So when these two sites, these two scatters is combined, we can derive results. Like you can see this graph, quadrant, the blue population, red population, and green population. So these are the three population. The uppermost population, as you can see, once you will mix these two graphs, you will show the green population showing higher granularity. That means higher side scattering, and higher forward scattering. That means larger the size. So the blue dots representing the amongst three, the larger size and larger complexity. Red dots representing moderate, medium size and medium side scattering, medium forward and medium side scattering. That means the medium size and medium complexity. And the green population showing in the graph, that shows the lowest amount of side scattering and lowest amount of forward scattering. That means among these three, this is the smallest in size and less in complexity. So in another example, how we can read the results from flow cytometry data. For example, I want to check the blood, different kind of blood cells in my blood. So I just draw my blood and test it with the flow cytometry and see what are the population in particular that sample, what are the neutrophils and monocyte and lymphocyte in that particular sample. So this represents the actual results. These neutrophils, because these are the large size molecules, they shows the large, they showed the higher side and forward scattering. So they come it just like it is shown here. Then similarly, monocyte is it literally less complex in comparison to neutrophil, but more complex in comparison to lymphocyte. They come in between the blue population and the green population. And lymphocyte, obviously, they come just near the X and Y axis, where the low forward scattering and low side scattering occurs. Similarly, that is second picture is not actually its replication, it is another picture. Why I have showed this picture here? Because we can also do the analysis of cell viability. Like if you do the assay with using dead cell, live cell, necrotic cells, apoptotic cells, you will find just similar this result similar to this result. Like dead cell will come in one quadrant, necrotic cell will come in next quadrant, apoptotic cell will come in one quadrant, and live cells will come in this quadrant. So on the basis of this, you can differentiate a population, how live cells you have in particular sample. So these kind of analysis also you can do. So first results is on the basis of their size and complexity, and second result example is on the basis of their viability. Similarly, this is the actual result experiment, like one who wants to see the T cell population, how much CD8 cells and how much CD4 cells are present. They have used different fluorescent dyes, for instance, and they found that 59.23% T cells and CD4 cells is 31.41%. So that's how you can, quantify the cell by using flow cytometry, or using the different phenotypic characteristics by you can differentiate the which cell population is CD4 cell or which cell population is CD8 cells. Even you can do furthermore, like CD4, the second picture, CD4 T cells, like how many different CD4 types, CD469 positive, CD469 positive, the different CD4 T cells, then different populations, how much different fluorescently tagged cells are present. You can also do the further analysis with the help of different detectors. Now we comes on to the major applications for what we have done this webinar, how important this flow cytometry technique in present scenario. Flow cytometry has, numerous application in modern healthcare research, including some of them I have summarized very briefly, because it comprises, I think, almost all, but very briefly and very neatly, because you can remember these things. So first is immunophenotyping. That is the very major important. How immunophenotyping, what it does mean. Flow cytometry is used for immunophenotyping, like means characterization of different immune cells. Which cells are present in particular cell population and which cell is not present. So it is very important. By analyzing the expression of different cell surface markers, we can identify the different cells population and track these changes populations during the immune response or disease progression. So by doing immunophenotyping, we can analyze the disease progression, we can understand the immune response, we can see what particular immune response is generated for what particular condition, and what are the different cells showing different kind of phenotypic characteristics or expressing a different kind of protein. We can measure by the help of flow cytometry. The second one is diagnosis of hematological disorders, specifically cancers. Flow cytometer is essential tool for diagnosing and classification of hematological malignancies, just like leukemia, lymphoma, and myeloma. And why it is important? By analyzing this expression of cell surface markers, researchers can identify the abnormalities, and they can distinguish between the different types of blood cancers. Actually, what type of blood cancer is present in particular patient. So what kind of medication we can provide for them. So it is very important to diagnose early. If it is early diagnosed, it can be treated better. Third one is monitoring of minimal residual diseases. Minimal residual diseases means what? Like if patient is taking medication for particular disease or some cancer or malignancy, then the doctor wants to check whether a person is recovered fully or not. Sometimes fluorescence microscopy or other microscopic techniques is not capable enough to identify a small trench of this particular, malignant tissue or something like that, this carcinogens. But this flow cytometry can easily detect this residual cancer from that particular patient who are on medication. So this is very important for the physician. They can check properly and continue the medication or stop the medication as per the requirement for this particular patient. So by identifying quantified residual cancer cells, physicians effectively treat the patient accordingly. Then the most important function is drug discovery and development. These are also important, but this is also very important. How it is important in drug discovery? Flow cytometry is used in drug discovery by screening several compounds. Because why we are using we are using drug for stopping a particular pathogen, microorganism, or inhibiting our some protein which is not desired for us. So we want to inactivate a certain thing, not our whole body. So you want to see whether the particular drug or particular molecule is really, causing damage to our cell. So we can do the cell cytotoxicity assay. We can see our cells are particularly viable or survival is proper. So we can do the survivability assay, survival assay, cell survival assay. So these things can be done with the help of flow cytometry. So drug discovery, in drug discovery, these things are extremely important, and these things can be achieved, these objectives can be achieved by using flow cytometry technique. The next is microbial analysis. So microbial analysis is also very important. As we see this COVID-19 pandemic, you have seen this COVID-19 were present everywhere. So it is not necessary or not possible to do tests everywhere. Everywhere you have to set the lab and all and do analysis for each and every sample. But flow cytometry, you can do analysis for different samples within like microfluidic samples, microfluidics lab on chip. It is also developed by recently by the researchers. They are doing to implicate this principle on point of care test, just like PCR. This is ongoing research, but it will take a little bit time. But this is very effective. So you can see the actual microbial analysis in which community. Yeah. So in which community, what kind of microbial flora, what kind of bacterial, viral, and fungal load is there. So you can determine the population. You can quantify the number of these pathogens. So this kind of analysis can also be done by the flow cytometry. So these are the major applications, but I have summarized these application into the two parts. Like one is a research laboratory and second is healthcare and diagnostics. So it's just the extension which I have just discussed before in last two slides. So first I'll go for the first application of flow cytometry in scientific research in day-to-day life. If you want to pursue the PhD or something like that, then what kind of experiment you are expected to do in your research lab. First I'll discuss those. Just like one first is intracellular antigen expression. It is very simple because you want to see the gene expression, you want to see the role of particular gene, what is the role of that particular gene. If you have cloned that gene, you have expressed that gene, and you want to see the how much what is the population of cell is expressing these genes. These kind of expression, antigen expression, you can see with the help of flow cytometry. Even that is why it is important with, in comparison to fluorescence microscopy. The expression level of a protein, like more than 100,000 individual cells, can be measured and visualized within a minute, which is not possible with the help of fluorescence microscopy. So it is very useful in terms of quantification. Number two is cell cycle analysis. Like you are doing like drug discovery or kind of related some molecule study, then you can see, you can do the cell cycle analysis. You can check the particular molecule in which stage is it is binding or it's suppressing or inducing. These kind of experiment is done by the laboratory for cell cycle analysis. So it is apoptosis. So this is also very important, whether cells are dying or not, what are the phenomena within the cell population. So you can do these apoptosis analysis with the help of flow cytometry. And important note is that the combination of this flow the apoptosis, like different dyes as propadium iodide is used, these dyes, the fluorescence dyes, will also give you a signal. That means what is happening inside the cell, how the cell is ruptured and not. And along with this, the forward scattering and side scattering will also give you a signal. Like low forward scattering and low side scattering shows that cells are in decline phase, they are in the death phase. So combining these things will give you a complete results, what kind of status in that your particular population. Next is cell proliferation assay. You also want to see in lab what kind of cells are proliferation, what kind of cells are being proliferated, what are not proliferated. So these proliferation assays can also be done by cytometry, flow cytometry. Then intracellular calcium flux assays. Calcium is very important for different phenomena, like how cells responds to stimuli. So different kind of physiological properties. If you want to explore these kind of experiment, you have to perform in your laboratory. So these are the important experiments you can do by flow cytometer. Now, the second one is the application in healthcare research and diagnostics. How this technique is important in healthcare research, and this is being, being important not nowadays, but in recent past. Because why? The instrument is very, sophisticated right now, because it the different changes and the innovation has make it possible to become it cheaper and more readily available, and simultaneously, because you will be surprised that like monoclonal antibodies and this FEX technology started simultaneously in 1970s, but monoclonal technology has, has a great role right now in comparison to flow cytometry. But flow cytometry is also giving attention now, because by use of different monoclonal antibodies, if combining these two things, flow cytometry along with monoclonal antibodies like fluorescence activated cell sorter, helps us to identify an n number of variety of things in our diagnostic era. So this is important in our healthcare research and diagnostics, like the direct advantages in current scenario, like diagnosis of leukemia, lymphoma, leukocyte surface antigens are measured in neoplastic cells. Then detection of minimal residual whatever I have discussed earlier, like the specifically CD13 and CD19, CD34 are measured in blood bone marrow. If these are upraised, that means the disease is not particularly cured. The hematopoietic progenitor cells enumeration, you can do this by using CD34 molecule surface marker. Histocompatibility cross matching, it is also very important when you are doing grafting or transplantation. So helatyping or histocompatibility matching is very important, and you can do by the help of flow cytometry. The post-transplantational monitoring, which I showed earlier, the level of circulatory CD3 positive cells, how much CD positive 3 cells are available in the cell, variety of antigens is measured. You can do this by the help of flow cytometry. Diagnosis of immunodeficiencies, you can also do this by help of this flow cytometry. If CD4 cell count is lower, CD8 cells are lower, then you can say that particular person or patient is deficient in immune response or immune cells. Then the microfluidic flow cytometry is also used for blood-based biomarker analysis. Ultimately, flow cytometry is not always diagnostic on its own, and it is most useful together with hematology, cytology, histopathology, and polymerase chain reaction, because that will supplement or complement that particular results you will find in flow cytometry. So along it will be difficult, because the result interpretation and some limitations are there. You should have proper sample injection and so many things. So if you merge these things like hematology, cytology, histopathology, and polymerase chain reaction (PCR) things, then you will get a very strong results, whatever you want to explore. So in respect to healthcare system, this environment is also very important. So with the help of flow cytometry, you can also do the environmental monitoring. Like on the basis of, we can also estimate the pollens, different the flagella, spores, and algal, cyanobacteria, different environmental organisms. We can measure and we can understand how environment is correlating with the immune system. So which is infectious, which is pollutant also. We can also measure the pollutant, because these are also a chemical molecule. So with the help of flow cytometry, we can identify the different particles, we can quantify them, we can do the morphological and physiological analysis. So flow cytometry is not only restricted to the laboratory for the using only the clinical aspects, it is also important for environmental aspects also. So which is the connecting link between healthcare and the environment. So in conclusion, flow cytometry is a powerful tool in healthcare research that is used to analyze the physical and chemical characteristics of cells and particles. And with the help of flow cytometry, we can also get the valuable information about the underlying biology of disease, as well as the potential targets for treatment and therapy. Thank you. Any questions?
Thank you, Dr. Arpit.
Yes, sir.
So now it is open for this question. Any questions?
Yes.
Anyone?
Anyone? Oh, somebody asked, I think, how can we analyze the drug-bound nanostructures? So I think your question is not very clear because our objective is, what is our objective? What we want to, analyze? A particular drug effect or the structure? Because if we want to analyze the particular drug effect, we want to see the cell is dying or not. So we will see the cells are dying, so we will do the cytotoxicity assay or an exin assay or apoptosis assay, whatever the we are predicting. So we are not finding the nano structure. We are just finding a particular drug is effective or not. Am I right? If you are asked to something else, then let me know. And somebody else.
Sir, thank you, sir. Sir,
Yeah. Yes.
myself, Dr. Kumar.
Yeah. Somebody has asked me, can we detect antibodies and quantification? Yes, we can do. But we have other techniques also, like the ELISA and other antigen antibody reaction assays. But in this technique, we can also do these things. But if it is really required, so we can do this. Yeah. Yes, infectious. Yes, we can also use in infectious diseases. For example, as I want to see the diarrheal pathogens. If you want to see the community in water source, water body, or in pond, you can see the analysis, infectious disease. What is the particular pathogen is present in that particular, sample. So these kind of, screening assays can also be done by flow cytometry. Oh, then what is annexin 5? Annexin 5 assay, you can see in detail further. That is, is used for the apoptosis analysis. What happens if a cell is dying? It shows the annexin 5 assays. Okay. So annexin 5 is used for apoptosis assay.
Anyone? Any other questions?
Hexa is a dye. Dye, we are using different fluorescence dyes. So on the basis of it, see different dyes, different fluorescence spectra is there. So these are the dyes.
So another question. Used in the dark.
It is useful for all.
Sir, someone is messaging for this Dropbox.
Yes. Sorry. Okay. I have one more question here. Like somebody asked, this technique is used for all samples, or there are some sensitivity. So you should be wise enough. Which sample we will go for this FAX? Which sample will go for the fluorescence microscopy? Which will go
Any
for
Catherine's
ELISA?
medicine shop
Like
अच्छी थी हो? So there's a bond that's here.
Excuse me.
I get it. Thanks.
Your voice is not
Yes.
clear. So my answer is that how sensitive it is. It is extremely sensitive because it, depends on the light scattering of light and hydrodynamics. So if it's a atom, it will give a different spectra. But when it is important to do, we have to decide. So it is very sensitive, but we have to do in a proper manner. It should be proper channelized. Otherwise, the interpretation will be difficult because we say like if one experiment fails, that if it does not give the desired result, it means it does not giving result. It is giving result, but it is not giving desired result. So it is sensitive, but you have to understand in which way you have to use this technique.
Any more questions? Okay. I think so, no more questions to anyone.
I think yes.
So we already share the feedback links. So everyone to be filled to this feedback form. Again, good evening everyone. So it has been such an honor to the part of this wonderful webinar events. On behalf of the organizations, I would like to extend my heartful grateful to the extended guest, our Dr. Arpit Shrivastava sir. Thank you, sir. It is a night imaginary task. I learned a lot. I sincere thank to my teammate also. Devasis, Amrit, Sriram, Susri. Their tireless effort to this effort to completion to these events. And finally, I would like to thank all these present here to making the times to be with us today and helping to making the event grand success. Thank you, Arpit sir.
And I would like to thank once again for inviting me and providing this such a great opportunity to interact with these students and you also. So I would really like to thank once again.
It's our honor. So
Okay.
thank you very much, Arpit sir. So
Okay.
hope everyone should understand to this what is the flow cytometry to use to this healthcare research perspective things. And this webinar only for this awareness of the students or professionals. Those are used for these tools and techniques of this flow cytometry, and they are further implemented into the careers. I'm really appreciated, sir, for your valuable time given for the times to all these participants. More than 140 or 150 participants to join today, and they are learning to this your thoughts. I'm really grateful to and all these participants. To
I
their,
hope
good
this presentation will be helpful for them.
Definitely. I think I learn a lot for this webinar. Hopefully, everyone should be understand to this nice idea and concept and tools of this webinar slides. I really love a lot to this things even. So, participants, please fill to this feedback form. So, accordingly, we will release to the certificates tomorrow onwards. And I'm really thankful to everyone, to anyone to contact to this officer. So I will put into this mail ID to this Dropbox. And any of the questions, if you have, so you will say to the mail to the officer. He will be when he will free. to reply your message, your query message.
Yeah, definitely.
Very
I will respond you. If anybody wants to ask me anything, you can ask me. Feel free to ask me. Whenever I'll get time, I'll definitely respond you.
So he is a nice personality, and I saw the LinkedIn. I said to the LinkedIn ID also to the officer. So and everything a lot to the officer. We will end up the sessions. And finally, I again thankful to officer and all these participants. Kindly fill to this form, and we will.
Thank you very much.
Thank
Thank you.
you. Thank you. Thank you.
A nice experience
sir.
with all of you guys.
Again, we will be trying for the another events to the webinar, new topics with officer. I am planning for the next month or next month this or up to June or July sessions. So again, you will meet with the officer to the new topic when he was free to the busy schedule. So he will be with us to the next nice interesting topics. So
Somebody
till
is
then,
asking, can I share this presentation? So I will share to you. So if anyone wants to see this presentation, you can, provide them. So I'll send you. Okay.
I think I drop a mail, your mail ID to the Dropbox. If
Okay.
anyone interested to the mail to your
Okay.
personal email, so we will try to.
Okay,
share.
then fine. Okay, I will respond them. Okay.
So that is a great for that.
Okay.
No. Okay, I will say to your mail ID. They are sharing for their mail ID.
No. You have to, clarify them once again. So maybe.
Okay. Okay. Okay, Arpit sir, this is a nice talk, and if you are free, and everything probably fill to this feedback form also. And finally, we will end up this session. Thank you very much.
Okay.
To
Thank you,
join
sir.
us to this event. Thank you very much. Thanks.
Okay, thank you.